So last Friday we talked about the three different types of solutions. We talked about isotonic, hypertonic, and hypotonic. And if you understand the prefixes of these words, you'll really help you understand what they mean. The prefix iso kind of means the same or equal. Like an isosceles triangle has three equal sides to it. The prefix hyper refers to like greater than or more. Like when you're hyper, you have a lot of energy. You have more energy than normal. And then the word hypo refers to less. Like hypothermia, your temperature's lower than it should be. So in terms of solution, we usually use the words hypotonic, hypertonic, and isotonic to describe an environment that the cell is placed in. If the cell is placed in a hypotonic environment over here, that means there's less solute in the environment than inside the cell itself. And what happens is that water tends to move in, tends to rush into the cell, causing the cell to swell. Yes?
Is there a possibility of this being not only within cells, but having it be within an environment we live in?
Sure.
Or is it only
Yeah. It's a way to describe any solution. Like if you were to swim in salt water, that'd be a hypertonic solution compared to you. But you are made up of cells, so. But if we put like a bag full of water, like fresh water, into a bucket of salt water, the salt water would be hypertonic to that bag. Just a way of describing the solution in terms of how much solute is dissolved in it. Okay, so they're more of comparison terms than anything. If we place a cell in a hypertonic environment, that means the environment has a higher solute concentration. Water tends to rush out of the cell shrivels up. Whereas isotonic environments, water moves in and out at equal rates. Cell does not change size. Now we talked on Friday, what is the danger of putting an animal cell, like a red blood cell, in a very hypotonic environment, kind of like distilled water? What could happen? It could burst. Okay, it could rupture the cell membrane, the cell would leak out, and the cell would die. Why don't plants undergo this? Why does it not happen in plants, Paige?
Because they have the cell wall.
They have the cell wall to protect them. Good. Who remembers the term used when a cell bursts because of its cytoplasm filling up with too much water? There's an actual word we use to describe that. Alex? Nope. That's this word when the cell shrinks. That's plasmolysis. You got that one. Kevin? Cytolysis. The word lyse means to break, to split, and it's the cytoplasm breaking the cell membrane. Cytolysis. Here's an actual picture with a microscope. You guys will get to do this in lab one day. Where we have normal plant cells. You can see the green chloroplast located here. And these are plasmolyzed cells. You can see that the cell membrane has been pulled away from the cell wall, and it kind of took all those chloroplasts and bunched them all up because the cells lost water. Basically, all I did was I added some salt water to the slide, and the cells basically shrunk. But notice the cell walls stay pretty much the same size. The cell walls don't change that much because they're very rigid compared to the cell membrane. So what does this have to do with cells? Well, cells live surrounded by fluid. Maybe except for the top layer of skin cells on your body, all your other cells are surrounded by some kind of fluid. And cells need to be able to take nutrients in, export materials like waste out, so they have to be able to transport things across their cell membrane. Things have to be able to move across that membrane in different solutions in order for transport to take place. So we say that the cell has a semi-permeable membrane. Let's look at this word, semi-permeable. What does semi refer to? Well, it doesn't have to be half exactly, but a portion of it, not a complete whole. And what does it mean to be permeable? To penetrate, to pass through. So semi-permeable means the cell allows some things to penetrate through it, pass through it. Other things it won't. That's pretty good for our cells. We don't want everything to move in or out because then the cell would just leak out indefinitely. So things that can move pretty easily are very small nonpolar molecules. Again, when we talk about nonpolar, we mean no charge. They have no positive or negative charge to them. So they zip right through the cell membrane like oxygen is nonpolar, carbon dioxide is nonpolar. They're very small molecules. They fit easily through the cracks of the phospholipid bilayer. They don't have a problem getting in or out. Polar molecules, molecules with a charge, including water, do not pass easily through the membrane. Even water can be slowed down because it has a charge. So a polar molecule has a charge to it. That hydrophobic region of the phospholipid bilayer, the cream part of our Oreo cookie, prevents molecules that have a charge from moving across. You have a hard time getting past that phospholipid bilayer because of the hydrophobic section that's in the middle. Then, of course, very large molecules like big proteins, big carbohydrates, do not easily move through it because they're just too big. They can't fit through the cracks. It's like trying to get an elephant to walk through this classroom door. It's just not going to happen. It's just the door's too small. Okay, so there are some molecules that cells need that are just too big to fit through. And they have a hard time getting in and out of the cell membrane. But the cell has ways of moving these materials through because the cell needs all of these types of materials. So before we move on, I want to do a little brain break, see what you remember from what we just talked about. I want you, either in your notes or on a piece of paper, I want you to draw a plasmolyzed plant cell. And in your drawing, include a cell wall, cell membrane, cytoplasm, and chloroplast. All right, quick energizer. I want you to stand up. Stand up. All right, transportation processes. There are two main types of transport that we're going to be talking about. Okay, transport can be broken down into two major categories: passive transport and active transport. Passive transport is the movement of substances across the cell membrane without the input of cell energy. The cell does not need to use any energy for passive transport. Whereas active transport requires cell energy. Now, we haven't talked about energy yet, so right now, energy is still kind of just an unknown thing to us. Well, some of you maybe, but I want you to write down your notes. The cell uses a molecule called ATP as its energy source. Think of ATP as kind of like money. When you guys need to buy lunch, you spend money. When you guys buy new clothes, you have to spend money. You go to the movies, you spend money. Okay, it's how you get things done. Okay, money, it takes money to do things in this world, in this society. Well, think of ATP as like the cell's money. When the cell needs to build a protein, it must spend ATP. When the cell needs to move around, it must spend ATP. When the cell has to undergo active transport, it has to spend ATPs to do that. Okay, so a lot of times energy, or ATP, is referred to as cellular currency, the energy currency of the cell. The cell needs to use it to get things done. And active transport is one of the uses that ATP has. Yeah?
Can they ever gain ATP?
Sure, we'll talk about how the cells make ATPs and make money, basically. We'll talk about that in a later unit. So focusing on passive transport. There are many types of passive transport. We have diffusion. Osmosis is a type of diffusion, and so is dialysis. And then we have facilitated diffusion. Both diffusion and facilitated diffusion do not require energy do not require the expenditure of ATP by the cell. So the cell gets to do this stuff for free. Costs no money for the cell to do this. Let's first talk about diffusion. When we talk about diffusion, we always say it moves down the concentration gradient. That's an important term there. Down a concentration gradient. Think of like a hill. Okay, if you start up on top of the hill and you wanted to roll a ball down the hill, will that cost you any energy? No. What takes care of it for you? Gravity. So things, on this earth naturally, will go down with no use of energy. Think of that as being down the concentration gradient. Except for the top of the hill is a high concentration of molecules, and the bottom of our hill is a low concentration of molecules. Molecules want to move from high to low just naturally. It's just the natural way things work. Okay, I'll show you an example. Anybody notice a different smell of the room today?
Yeah.
Okay, because I plugged in this air freshener today. All right, because we're talking about diffusion. This air freshener works on diffusion. Okay, if you take the little cartridge out, in this little plastic tube are a bunch of fragrance molecules in a really high concentration. Out in the room, it's a low concentration. And so they naturally want to diffuse. I could even leave it out on the desk and not have to plug it in. And they would naturally diffuse out into the open room because there's less concentration of molecules out here, and these move from high to low. All right. Same thing can be said for like those. Anybody have Axe body spray? That stuff works the same way. Perfume works the same way. Okay. Fragrance molecules move from high to low concentration. Just the way things work. Okay. So when we say down the concentration gradient, we're basically saying from high to low concentration. So a concentration gradient is the difference between the concentrations of a particular substance in one area and its concentration in the adjacent area. So in our little fragrance molecules, air freshener example, this is a high concentrated area. Out in the room is a low concentration area. They have to be adjacent. It's not like these molecules are going to magically move from here into the front office, because it's not adjacent to the front office right now. Okay? Eventually they'll make their way there, maybe. They'll move from this room into the hallway, then down the hall, then make their way there. But by that time, they'll be so spread out, you probably wouldn't smell them anymore. There wouldn't be many molecules left to smell. When molecules have dispersed evenly, there is no concentration gradient. All right? Over time, these will spread out enough that the whole room will have about the same concentration in every area. It's not like all of these molecules are going to eventually make their way all the way to the back of the room and recongregate and get back into a nice, tall, tight little cluster and be highly concentrated again. They spread out. Eventually, these molecules will leave the school. They'll leave Lake County. They'll just spread out into the world, and you won't be able to detect them anymore because they're so spread out. But once everything is even, there's no concentration gradient anymore, diffusion stops. Equilibrium is reached. So if you have no high and low, there's no movement. It's like a ball won't naturally just roll across a flat table because there's no difference in elevation. Okay, so if there's no concentration gradient, there's no more diffusion taking place. Some factors to consider. The rate of diffusion, how fast diffusion happens, depends on the characteristic of the membrane. The membrane could be very permeable, could not be very permeable to whatever molecule you're looking at. Also depends on the size and temperature of the molecules involved. Now, when you guys think of temperature, you probably think of how hot or cold things are, right?
Yes.
That's the everyday person's understanding of temperature. But temperature really is how fast the molecules are moving. That's what temperature is. It's just how fast the molecules are moving. Really, really fast, really, really hot. Really, really slow, really, really cold. So, back to our fragrance molecules. To get these to move out the room faster, what do?
Put them in the air freshener.
Put them in the air freshener. This plugs into the wall. What do you think that does? Heats it up. Okay, this basically gets a little bit warm and moves those molecules throughout the room that much faster. Molecules moving faster, they'll diffuse faster. And then smaller molecules diffuse faster just because they can kind of get through the air faster. They can zip through the cell membrane quicker because they're smaller. Kind of like our proteins moving down the gel. The smaller ones moved faster because they could fit through the smaller places. And then the greater the concentration gradient, the faster the diffusion. Think about our hill example. The steeper the hill, the faster you go down it. There's more of a difference. The faster diffusion takes place. So as the concentrations start to even out, what do you think happens to the rate of diffusion? Does it speed up or slow down?
Slow down.
Slows down. Eventually it stops altogether. So here's our definition. You don't have to write the definition down. What's important, though, is what molecules can easily diffuse. Oops, let me go back. Oxygen and carbon dioxide diffuse across the cell membrane because they're small enough. They don't have a charge. Move from high to low concentration. So things can easily slip past those phospholipid bilayers. So we get oxygen into our cells and CO2 out of our cells by simple diffusion. Does that require us to use energy? No. It's a free service. Okay, here's a short video clip on diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
So diffusion does not have to occur across the cell membrane, but in terms of how cells move things in and out, diffusion will be across the membrane. Okay, but diffusion also happens in just beakers of water. So here's another picture, just an image of it. So if we have a lot of high concentration molecules outside the cell, this yellow thing represents the cell. Eventually those molecules will diffuse in, as long as they can fit across the membrane, and even themselves out and be spaced out inside the cell. Yeah?
What happens if they can't fit across the membrane?
Then they can't diffuse. They just stay at a high concentration outside the cell. Just like if an elephant was outside our door and really wanted to be in here,
Crash through the wall.
well, I guess if they crash through the wall, if they crash through the cell membrane, the cell would be in trouble.
Could riot.
Now, osmosis is a special form of diffusion of water, and it's water across a semi-permeable membrane. So water, flowing across the floor is not osmosis. All right, water moving across a membrane is osmosis. Yes.
I've seen on water bottles that said
Reverse osmosis.
Yeah.
Yeah. They use energy to move water against the concentration gradient from low to high. But yeah, that's reverse osmosis because it's moved across. It must be moved across some kind of membrane. I'm assuming the membrane's like a filter. probably just a really fancy way of saying filtered water. Yeah. Not even that, because that sponge is still, it's not across the membrane. You have to be moving like into or out of the cell, basically. Though there are artificial membranes we'll talk about, but not even a sponge. A sponge just holds the water in like little air spaces that are inside the sponge. Occurs when the two concentrations of solutions are on two sides of the semi-permeable membrane are different. Water moves down the concentration gradient, moves from solutions of higher water concentration, low solute, to solutions with lower water concentrations, high solute. So this goes back and explains why a cell loses water in a hypertonic environment and why a cell gains water in a hypotonic environment. But we'll save that for tomorrow because that's all the time